Hello my dear students, today we are going to unfold the architecture of the plasma membrane through the lens of the fluid mosaic model, one of the most remarkable concepts in modern cell biology. This model revolutionized our understanding by showing that the plasma membrane is not a rigid wall but a flexible, dynamic and mosaic-like structure where lipids and proteins move freely to perform vital functions. It beautifully explains how the membrane maintains stability while allowing fluidity, communication and selective transport. So let's dive into the fascinating world of the fluid mosaic model and explore how it reveals the true architecture of the plasma membrane. Fluid mosaic model, it was proposed by S. J. Singer and Gert L. Nicolson in 1972. Further, it was revised by Nicolson in 2014. The key features of the original fluid mosaic model, which was described in 1972, is following: lipid bilayer, which is composed mainly of phospholipids arranged in a bilayer within the cell membrane. The hydrophobic fatty acid tails face inward, while the hydrophilic heads face the outward. It acts as semi-permeable barrier, maintaining cell integrity and regulating transport. Fluidity of the membrane: lipids and proteins move laterally, giving flexibility. Unsaturated fatty acids prevent tight packing, increase fluidity. The cholesterol prevent excessive rigidity or fluidity and stabilizes the membrane structure. Mosaic of proteins. Integral proteins span the bilayer and function as channels, transporters and receptors. Peripheral proteins, they are loosely attached on surfaces, functions in signaling, anchoring and cytoskeletal linkage. Its arrangement resembles a patchwork mosaic. Membrane asymmetry. Inner and outer leaflets of the cell membrane differ in lipid and protein composition. Outer surface, glycolipids and glycoproteins help in cell recognition and cell signaling. The inner surface proteins link to the cytoskeleton. Selective permeability, it allows controlled entry and exit of molecules within and from the cell membrane. It involves passive diffusion or facilitated diffusion and help in active transport, which is driven by ATP driven pumps. Dynamic nature, membrane is flexible and adaptable. Its components, which are lipids, protein and carbohydrate, constantly move, rotate and rearrange within the cell membrane. Why plasma membrane is called as fluid and mosaic? Fluid nature, lipids and proteins move laterally. There is no strong covalent bonds. The unsaturated fatty acids and cholesterol regulate the fluidity of the membrane. It ensures signaling, transport and membrane fusion. Mosaic nature, membrane shows a patchwork of proteins, lipids and carbohydrates. Proteins perform transport, signaling, recognition and structural roles. Movement of the phospholipids. The first one is the rotational movement. This is the rotational movement. Phospholipids spin around their axis, that is rotational movement. Lateral movement, phospholipids can shift within the same leaflet, that is lateral movement. This is lateral diffusion, or it is rotation or flexion. Transverse movement, if they change their bilayer, flip-flop movement, it is very rare and it exchange between leaflets. Specialized enzymes for flip-flop. Flipase, which is located in the outer cell membrane and moves into inner leaflet. The example is phosphatidylserine. Floppage, it is usually present in inner leaflet but it moves into outer leaflet. The example is phosphatidylcholine. And scramblases, it is responsible for bidirectional exchange. It is ATP independent process and activated by calcium 2 plus ions. Factors affecting membrane fluidity. Temperature. Low temperature, phospholipids tightly packed when it is less fluid. In a high temperature, when it is more spacing, more fluid. Saturated versus unsaturated fatty acid. Saturated fatty acids are straight chains, tight packing and less fluid. Cis unsaturated fatty acids are kinked. They prevent tight packing and it is more fluid in nature. Trans unsaturated fatty acids, they are straighter and becomes rigid and they are less fluid. Cholesterol, they maintain fluidity at both high and low temperatures. It prevents too much packing in cold and too much separation in heat. This is the membrane fluidity graph versus temperature, and this is the condition with high temperature when cholesterol is without cholesterol or in presence of cholesterol, and this is the condition at low temperature in presence of cholesterol or without cholesterol. This is the role of unsaturated hydrocarbon tails with kinks in the membrane fluidity, and if saturated hydrocarbon tails are there, membrane become viscous. Revised fluid mosaic model, which was proposed by Nicolson in 2014, refined lipid protein and protein protein interactions, greater emphasis on complex interactions in signaling and transport, membrane microdomain or lipid rafts. Specialized regions enriched in cholesterol and sphingolipids, they play critical roles in signaling, trafficking, and protein sorting. Membrane asymmetry reinforced. Asymmetry in lipid and membrane distribution is essential for recognition, signaling, and membrane stability. Dynamic membrane behavior, it highlights continuous motion of lipids and proteins. It emphasizes adaptability for cell function and survivability. In summary, the fluid mosaic model in 1972 explained the plasma membrane as a dynamic lipid bilayer with proteins form a mosaic structure. But in 2014 revision integrates new insights including lipid rafts concept, asymmetry and detailed molecular interactions of the different components of the cell membrane, showing the plasma membrane as even more organized, complex and dynamic than originally proposed. If this video helped you understand the architecture of plasma membrane through the fluid mosaic model, don't forget to like, share and subscribe to the channel for more insightful biology lessons. Stay curious, keep exploring, and I will see you in the next class. Thank you.